This is for cellular respiration. For microbiology, we tend to focus in the electron transport chain. I will make a different video for glycolysis, transition step, and the Krebs cycle. But for now, so we can know there's cytosol here, and different functions occur here. And then in the mitochondria, we're going to speak about the outer membrane, the intermembrane space, and the inner membrane. The intermembrane space is just you could think of that as inter, meaning in between, so that's just an in-between space, and then the matrix of the mitochondria. So when I speak of these areas, just recall the mitochondria, or the cytosol, recall the cell. Now, rather quickly, for cellular respiration, in eukaryotes and prokaryotes, glycolysis occurs in cytosol. For eukaryotes, the transition step occurs in the mitochondria, specifically the matrix. Recall the image. Krebs cycle in the eukaryotes occurs in the mitochondria again, but in the matrix specifically. The electron transport chain occurs in the mitochondria, specifically in the inner membrane. So recall these areas. Matrix, we have the Krebs cycle, transition step. The inner membrane, we have the electron transport chain, and the cytosol, we have glycolysis. For prokaryotes, glycolysis, transition step, and Krebs cycle occurs in the cytoplasm, and the electron transport chain occurs in the plasma membrane. So now, going further for counting ATPs, let's talk about the aerobic respiration versus anaerobic respiration and fermentation. I have check marks for everything that's considered that occurs. For example, in aerobic respiration, a check mark for glycolysis, Krebs cycle, and electron transport chain. 2 ATPs, 2 ATPs, 36 ATPs, 38 in total. Glycolysis has, in the anaerobic respiration, has 2 ATPs. For the Krebs cycle, has less than 2 ATPs, and in the electron transport chain, it has less than 36, for a total of less than 38 ATPs in total. Fermentation, the only thing that works is the glycolysis for 2 ATPs. Krebs cycle and the electron transport chain does not work. Perhaps you're wondering why the transition step isn't here. It is because, as you recall, cellular respiration, there's 0 ATP at the transition step produced. Okay, now going into the electron transport chain. If you recall the first image, we have the cytosol, we have the outer membrane of the mitochondria, the intermembrane space of the mitochondria, meaning the in-between, the inner membrane, and then finally the matrix of the mitochondria. For cytosol, we have it occurring here in the cytosol, glycolysis, and then we have the transition step occurring in the matrix, Krebs cycle occurring in the matrix, and the electron transport chain occurring in the inner membrane. For now, let's speak of two major terms: substrate level phosphorylation. I'll go into depth of what that means, but substrate level phosphorylation occurs only in glycolysis and only in the Krebs cycle. For oxidative phosphorylation, it only occurs in the electron transport chain. So now specifically glycolysis, substrate level phosphorylation. When you think of glycolysis, just say this term and then include this last term here. So for example, substrate level phosphorylation for ATP production by transferring high energy phosphates from a phosphorylated substrate to ATP, basically meaning ATP production by transferring phosphates from a phosphorylated substrate to ADP. Continuing on with glycolysis, in glycolysis, glucose is oxidized or loses its electrons to pyruvate, and NAD plus is reduced or gains its electrons, to NADH. If you want to be more specific, like using the whole cellular respiration, you could use it again. When you think about glycolysis, think cytosol, and think about substrate level phosphorylation for 2 ATP productions by transferring high energy phosphates from phosphorylated substrates to 2 ADPs. In glycolysis, 1 glucose is oxidized to 2 pyruvates and two Nad pluses are reduced or gains its electrons to two NADHs, if you want to be more specific. For substrate level phosphorylation, we have again the same definition. For ATP production by transferring high energy phosphates from phosphorylated substrates to ADP. In the Krebs cycle, acetyl-CoA, 2-acetyl-CoA specifically, if you want to be specific, gets oxidized or loses its electrons to 4 CO2s. And NADH NAD plus gets reduced to NADH, specifically 6 NADHs. FAD gets reduced to FADH2, specifically 2 FADH2. Okay, now the electron transport chain, we have a series of reactions occurring here, but to make it a little bit easier, if they ask you what is oxidative phosphorylation, then we're going to think of electron transport chain. Oxidative phosphorylation for ATP production by transferring high energy electrons from organic molecules to electron carriers. In electron transport chain, NADH is oxidized or loses its electrons to NAD+. FADH2 is oxidized or loses its electrons to FAD, and a series of oxidative reductions of electron carriers, basically meaning a series of electron carriers gain electrons or give up electrons. So there's 3 steps for that. We have the first step in the electron transport chain. We have high energy electrons transfer from NADH, high energy electrons, hence those 2 dots there, get transferred from NADH to FMN, flavin mononucleotide. So, and then also know that the first electron acceptor is the FMN. So when NADH gets reduced to NAD+, we have an H or hydrogen with 2 electrons that passes to the FMN. So it's unbalanced, so it needs another hydrogen to make it balanced. So FMN picks up a hydrogen from the matrix and then gives it to FMN. NADH is oxidized, loses electrons, it becomes NAD plus, and FMN is reduced or gains electrons. It becomes FMNH H2. So flavin mononucleotide plus 2 hydrogens, right? 1, 2, and then the electrons are there, just to remember that they're there. Then the second step in the electron transport chain system is that, and then hence the fuchsia color, we have the flavomononucleotide plus 2 hydrogens passes the 2 H pluses, so passes its to the 2 hydrogens to Q coenzyme Q, and while it does that, I guess you could say that it transferred to the intermembrane space specifically. That's why there's high concentrations of H pluses there. The FMNH2 also gets oxidized and becomes FMN again, because it gave up its 2 electrons and it also gave up its 2 hydrogens. So then the second part is that coenzyme Q grabs 2 hydrogens from FADH2, flavin adenine dinucleotide, plus 2 hydrogens from the matrix, and releases it to the intermembrane space. Boom. The final third step is a series of oxidation reductions. As we mentioned for this definition, cytochromes basically gains electrons and gives up electrons. So for example, the coenzyme Q gets oxidizes or loses its electrons because cytochrome B accepts the electrons, and then it goes to cytochrome C1, cytochrome C, cytochrome A1, cytochrome A3, and then finally the oxygen. Just note that the cytochrome A3 is the final electron carrier. So now that we have oxygen here, note that there is high concentrations of H pluses in the intermembrane space for the same reasons that we mentioned earlier. And then there is low concentrations of hydrogens in the matrix. So hydrogen naturally was going to want to grab from here, from this space, because of two reasons. There's the electrical chemical gradient, electrical meaning opposite attract, so that positive wants to go with negative, because oxygen is negative. And then also the electrical chemical gradient, so the chemical gradient or concentration gradient, meaning since there's a high concentration here and there's a low concentration here, it's going to naturally want to go down. So these are the two reasons why, due to oxygen, hydrogens go down the ATP synthase, 2 hydrogens, and fuse basically with 1/2 of oxygen, which creates 3 H2Os. For every 2 hydrogens that go down the ATP synthase, we make 2 ATPs. Just last thing, this area here, I guess you could say maybe like a protein. It's going to be called the NADH dehydrogenase complex. Dehydrogenase just basically means NADH becomes NAD plus, or NAD plus becomes NADH. That's all that dehydrogenase basically means. And then the cytochrome E BC1 complex is this complex here. Cytochrome oxidase complex is this last one here, and then the ATP synthase is right here.